Great. Thanks for inviting me to this, Nanda, and thanks for setting all this up. Like Nanda said, my name is Lyndon, and today's talk will be about my recent work. We just had it accepted to ICLR on shape metrics for neural representations. It's nice to be virtually back. I'm actually Canadian and went to McGill, so in a couple of weeks when I'm back for cosign, maybe some of us can meet up and enjoy Montreal together. It's been a long time for me. So everyone can see my screen okay, by the way? Okay, I'm going to take that as a yes. So the primary goal of the work that I'm presenting today is to compare neural network representations. Comparative analyses are fundamental to the scientific method. For example, comparative science allows us to do neural recordings in rodents and monkeys and tell government funding agencies that what we learn can be applicable to the human brain because of anatomical analogs. It's also the reason why we know which genes can cause disease in a sick patient versus a healthy patient. It's also key to ethology, where researchers try to quantitatively analyze and differentiate animals based on their behavior. In systems neuroscience specifically, comparing representations is also a familiar goal. Nico Krieges-Korte and colleagues introduced representational similarity analysis to compare object representations in monkey with human inferotemporal cortex. And more recently, Dan Yammans and Jim DiCarlo have sort of spearheaded efforts in using regression analyses to see how well deep nets can explain monkey neural and behavioral data. So what's important to note here is that these methods here are well suited to comparing just two networks, A and B, or explaining one network, X, with another network, Y. But in neuroscience, large collaborative efforts are now producing massive data sets comprising several dozen animals. Actually, at this point, I think it's even over 100. So what I'm showing you here in the diagram is a simple decision-making task where a mouse is trained to steer a little Lego wheel left or right, depending on a presented stimulus. So labs from around the world, from different institutions, trained dozens of mice to do this same standardized task. And they also recorded neural spiking activity from regions of the mouse brain involved with decision-making. The black lines in the brain at the top right show the probe trajectories, and colors denote brain regions of, looks like visual cortex, hippocampus, and in pink, thalamus. So key to this experiment is that the hypothesis is that neural activity is correlated and with task-related behavior, as well as with idiosyncratic differences between individual mice. So with emerging large data sets of many network responses calls for methods that are suited for sort of mass comparisons between brain areas and animals. So the outline of my talk today will be about representational geometry in neural representations and how to compare them. So first I'll talk about what representational geometry means. Then I'll go into formalisms of what a metric is, and then describe methods recently proposed by us called shape metrics on deterministic and stochastic neural representations. And then I'll finish it off with a fun little application I did with variational autocolors. So I hope that I have convinced you that we're kind of in the era of neuroscience and machine learning now, where experiments produce huge data sets from multiple animals or networks performing the same task. So here what I'm showing you are two matrices representing simultaneously recorded activities from neurons in two different networks. You can think of these as responses from two different animals, brain regions, deep net layers. Each column is a neuron, and each row is the population response to a given stimulus condition. So a fundamental question in representation learning and machine learning and in neuroscience is, given multiple networks that were doing the same task, how related are their neural representations to each other? These network matrices are high dimensional with the number of dimensions equal to the number of neurons. But for the sake of visualization, let's just assume that the responses each lie on some low 3D manifold of responses, where each row of the matrix representing the population response to a condition is plotted as a colored point in this space. So we have these like two Pringles chips for neural manifold 1 in purple, and a slightly warped and rotated green Pringle chip for network 2. So at this point, there have been many proposed methods to answer this question of neural network representation similarity. Those include the RSA and linear regression that I described on the previous slide, but there are also others like canonical correlation analysis, more recent ones like centered kernel alignment. One that's most similar to what we're doing is based on shapes and this idea of co-registering two shapes together with a Procrustes alignment. But as I mentioned before, most of these methods are good for comparing two networks with each other. Well, what if we have many networks? Ideally, we would like a tool that allows us to compare all networks in a single space simultaneously. And in this space, we should strive to have that our basic intuition of closeness, as in two networks are close together in this space, means that their representations are close. And this will enable downstream analyses like grouping and clustering of networks, and should have theoretical guarantees of correctness. One way to ensure this is the one that I'm going to focus on today, and it's by measuring dissimilarity distances that can mathematically be defined as metrics. So what is a metric? We want to formulate functions D that take as inputs neural representations, X, Y, or Z, and adhere to the following 4 axioms. The first is that a metric or a distance should be non-negative. This is very intuitive, but this already rules out measures like correlation, because correlation can be -1 all the way to 1. The second is equivalence. If the D of X and Y is 0, then we should conclude that they are equivalent to each other. The third is symmetry. Things like regression, for instance, are not symmetric. Deep net activations can explain a small population of neural data, but that neural data cannot explain the activations of a deep net. And finally, and perhaps the most important one when it comes to describing many neural network representations together, is the triangle inequality. And basically this says that the straight line between X and Z, red to green, should be shorter than taking the long way around of X to Y to Z. Now, the triangle inequality is particularly important when we have more than 2 networks. If the triangle inequality were violated, then we could simultaneously measure X and Z being close to Y, but somehow also measure that X and Z are very far from each other. And this already sort of breaks our natural intuition of what distance means. And this already sort of invalidates downstream analyses that try to group together similar representations using measures that violate the triangle inequality. And that just would no longer make sense. So our answer to that is to introduce what we call shape metrics. And first I'm going to go through metrics comparing deterministic neural responses, followed by the recent work that I did on stochastic extensions of this framework. So our method draws inspiration from the field of statistical shape analysis. Say we have a network that takes as input Z and spits out f of Z. We do this for many networks and collect the responses into a single matrix X. We do this for 2 different networks, and we want to treat each joint response as a high dimensional shape, i.e. the 2 Pringles. We want to rigorously quantify notions of distance between these two Pringles, these two shapes. And the idea is that the distance between these two geometric shapes should be independent of some group of nuisance transformations. So given an allowable set of transformations, we should be able to first align the two networks, and whatever remaining difference is the distance between them. So this T, this transform, formally must be in a group, which means that it must contain the identity transform. It must be invertible by another T in the group, and a bunch of T's must be closed under composition. Oh, and finally, T must be an isometry, like a permutation or a rotation. And these conditions together effectively make our method symmetric. So you can choose to either align I to J or J to I, and you should get the same distance. Does anyone have any questions so far? Okay, feel free to interject at any time. So concretely, we have two response matrices, I and J. And already at this point, the most common concern or question that comes up is, what if I record from monkey I and monkey J, or area I and J, and I don't have the same number of neurons? In other words, what if the dimensionality of these matrices is not the same? Well, for this, we allow for some feature map, phi, and this acts like a preprocessing function. And what this can do is basically any arbitrary nonlinear function, and it's up to you to decide as a experimenter. But for instance, if we have this mismatch of dimensionality, I can either set phi to append a bunch of columns of zeros to one, such to I, say, such that the dimensionality will match J. Phi can even just be the identity if they already match and you don't want to do anything. It can be an isotropic scaling. It can perform PCA on both the networks to reduce them to a common dimensionality. And it can even be nonlinear. But no matter what you choose phi to be, the resulting matrices are then aligned by a single global transform T in the group that we define, G. And whatever is left over after alignment is what we call the distance. Are there any questions? This is sort of like the heart of the method. So if there's any questions about maybe some intuition or concerns, please raise now. Okay. How is this similar to manifold alignment? Could you describe one or give one as an example? This sounds like it is very similar, and they possibly could be sort of complementary approaches or two different fields that use different terminology to describe the same thing. Sure, yeah. Yeah, one thing that people ask is if phi is so flexible, it can be nonlinear, then isn't it doing all the work in doing the alignment? Can't phi just do the alignment? And really, I think at the heart of it, that allows me to segue into my next slide, actually. Phi encodes hypotheses that we have about representational similarity. For instance, if I don't do anything and the dimensionalities match, and I just define my nuisance transformation group as rotations, and then I align, then this produces a Procrustes distance, or what is sort of classically known in the shape statistics literature. However, if we were to, for example, perform a whitening transform on X and Y before alignment, let me see, oh, there it is. Then it turns out that this yields something that is very close to canonical correlations analysis. And this would provide a distance that is invariant to any arbitrary linear map. So because shape metrics are a bona fide metric, we can analyze pairwise distances between multiple networks simultaneously. And this enables downstream clustering analyses and visualization with theoretical guarantees on correctness. What this plot shows here on the right is about 50 mouse brain areas during some recordings where mice were sort of marching around and watching a natural video data set. And by analyzing their representational similarity between these different brain areas with shape metrics, this allows us to plot all the networks into a common space, which we call shape space. We didn't coin this term. This is already a standard term in the field. So the procedure is you have these, say, 50 different response matrices. You compute a 50 by 50 distance matrix. Then you embed each network using multidimensional scaling. And here I'm plotting the embedding projection along the top two principal components, just for visualization. T can be, yes, T is a linear isometry, so it is a permutation or a rotation or something in that group. Most of the things that I'm showing here today, actually all the things I'm showing you on T will be a rotation. Thanks, Booyah. Right, so what I'm plotting here is the 50 different networks in shape space. So each point here is actually a network. It's not an activation space or in stimulus space. And the colors here, it's important to note, were added in post hoc. So the colors represent different anatomical regions of the mouse brain, and the method had no idea about anatomy or functional connectivity in the brain. But what we find is that this shows that shape metrics provides a nice unsupervised way to discover functional similarity between these different networks. So we can see all the blue ones are clustered together, all the pink ones, all the green ones. Are there any questions? So I think there are some scientists in the room. I think the first thing, especially an experimentalist will say, is that real neural responses are stochastic. Imagine now that I'm showing networks a stimulus, either some dark blue or cyan stimulus. And every time I show the dark blue stimulus, it elicits a different response. However, there is some conditional mean and conditional covariance about that mean. So notice here that in network 1 and 2, the white stars are the same. So that is to say the conditional means are the same in both networks, but the shape of the conditional distributions, the ellipses here, are very different. In neuroscience, these are referred to as noise correlations and exist in all brain areas across all species, as far as I'm aware. It's therefore important for us to develop a way to compare how stimuli are encoded in different animals, not just by their conditional means, but also taking into account their conditional noise. We need a notion of distance and an alignment procedure that takes into account noise in each response. And one natural way to do this, and is a very natural sort of stochastic extension of the deterministic shape metrics that I showed in previous slides, is to rely on Wasserstein distance. So ideas from the field of optimal transport theory. So imagine now you have two networks and their responses in 2D are purple and orange, responding to some same fixed stimulus. So each of them have a conditional mean and now a conditional covariance about that mean. If we model the responses as Gaussian, then the Wasserstein distance, here it's actually the 2 Wasserstein distance for aficionados, has an analytic form which nicely decouples into two quantities very familiar to experimentalists. The first term is simply the difference between the network conditional means. And the second is known as the Burris distance, which quantifies the difference in orientation and scale of the covariance clouds. Intuitively, you can think of Wasserstein distance as the amount of work it needs, or I would need, to move and transform one pile of dirt that is shaped like the purple density and transform it into the orange density, or vice versa. So this Wasserstein analytic form nicely splits this work involved with either moving the center of mass of the density plus the work needed to reshape one ellipsoid into the other. And it's worth mentioning here that when the covariances are equal, or when there is no noise, then the second term drops out, and we arrive at our L2 distance between the means, as I showed before. So this method is literally like a natural stochastic generalization of the deterministic shape metrics in a couple slides ago. And it's important to note that here we're modeling our densities as Gaussian. So your responses don't literally have to be Gaussian distributed. Yes, thank you for asking that, Colin. Yeah, it is mainly a approximation because it yields this nice analytic form, and because most experimentalists, or at least when I worked in a monkey lab, we would measure the conditional mean and conditional covariance to measure noise correlations. And these are just natural quantities that are typically always measured in these sorts of experiments. So we wanted to provide a tool that is just a natural thing for people to turn to and use immediately. We also, in a couple slides, I'll go through briefly another method that takes into account higher order moments. But thanks for your question. So to operationalize Wasserstein distance into our shape metric framework, imagine now I just have a single input to two separate networks, and this elicits a different response and different covariance for each one, as shown on the screen here. So our method aligns the two networks by considering now both the means and the covariance. And you can see this here denoted by the T in front of the mu J and on either side of the sigma J, where sigma is the covariance of J. And you can convince yourself the T has to be on both sides of the covariance here because the resulting covariance has to remain symmetric. So the idea is that we want to find a global or a single T that globally aligns one network coordinate frame into another. And we are now accounting for both means and covariances. And just as an aside, this optimization for T is highly non-trivial, and I go through details in the paper about it, or we can talk about it after if you're curious. Basically, you want T to always remain a rotation throughout optimization, and that is hard. So the previous slide showed you two networks' responses to a single stimulus. In the more general case, we're showing several stimuli and recording conditional densities in the responses to several stimuli. So the idea is we have this joint distribution for network one response to 5 different stimuli, and we want to transform or rotate network 1 into network 2 with T, as shown here, and whatever's left via Wasserstein distance is the distance between the two distributions. Does that make sense to everyone? Great. So to answer Colin's earlier question, whereas Wasserstein can be thought of as a sort of what's called a transport plan that maps one density into another with the least amount of work, in the paper, we also propose a different distance called energy distance, which describes a different transport plan that is the maximum entropy transport plan. Intuitively, it is the cross covariance between two aligned joint densities that is not explained by within network variance. It turns out that this is more sample efficient because Wasserstein is difficult to estimate when your dimensionality is really big, and because there's no sort of closed form solution in most cases, this takes into account higher order moments. It's very good for sort of empirical densities. Yeah, I won't go into all the details, but I just wanted to be clear that modeling your responses as Gaussian is not the only option. So you may have noticed that the Wasserstein is just the equal weighted sum of two distances, the covariance Burris distance and the means distance. And each term alone actually defines a metric. And the sum of any two metrics in general is still a metric. So to explore the contribution of means distance and covariance distance and their individual contributions to representational differences in geometry, we introduce a simple scalar parameter alpha, which ranges between 0 and 2, that sweeps a continuum of Wasserstein distances. That is, when alpha is 0, we're only looking at the Burris distance, like just differences in covariances. And when alpha is 2, it's, as before, we sort of get our deterministic shape metric back. And then when it's 1, we get our familiar Wasserstein distance. So to validate our stochastic framework, we generated 100 network representations. Here I'm showing you 16 of them aligned in a 4x4 grid, where all networks have the exact same conditional means denoted by the colors here. Going from top to bottom though, we isotropically scale the responses, and from left to right, we vary the correlation or the orientation of the conditional covariances. We then randomly rotated them and computed a 100 by 100 shape metric distance matrix, where rotations were chosen to be the nuisance transformation. And by taking this distance matrix and embedding it using multidimensional scaling, we can accurately recover the 2D organization of all the networks that sort of sensibly sweep along two different axes here, the scale and the correlation. Unsurprisingly, if we take our interpolated Wasserstein metric described on the previous slide, if we set alpha to 2 and only take means into account, since all the means are the exact same, we just get trash. We do not recover the structure in the data. And similarly, if we ignore the means and only take covariance structure into account, the plane up here in the top right kind of folds over onto itself, because you need the means to still sort of achieve global alignment in the representations. So by construction here, we are only able to recover the true structure of the data, the networks, by considering both the means and the covariances. And this slide is mainly to show that since most existing methods only consider deterministic geometry, none of them are able to account for, or none of them are able to recover the structure that we can by taking into account the noise. So for the final part of my talk, I'm going to sort of validate and scale up, show how we can scale this new stochastic metric to thousands of networks. And I'm going to be applying that to variational autoencoders, or VAEs. I'm sure many in this room are actually very familiar with VAEs. I know Colin is, but in case you're not, a VAE takes as input an image, and then a neural network then serves as a bottleneck. And for each image, it spits out exactly, by design, a Gaussian distribution. After which, another neural network lifts out samples from these conditional densities to reconstruct the input. These models are a perfect testing bed for us because the conditional responses are exactly multivariate normal distributions. And so using our Wasserstein shape metric to compare different networks is actually exact. It's no longer an approximation. And what I'm going to do is take about 1,800 different networks trained with different objectives and hyperparameters and architectures and compare them to each other. It's important to note that VAEs, for better or for worse, have become very popular tools across all science disciplines as a method of nonlinear dimensionality reduction. But when people plot these latents, when they dimension reduce their data, researchers typically throw out the covariance and only look at the conditional means. So here I'm actually retaining the covariance information to see how much it can tell us about the network and how it relates to different networks. So I computed distances between 1,800 VAE latent representations trained on a simple data set called D-sprites. Each one is trained using a different class of objective functions here in colors. The details are not important for this talk. But you can just think of them as different objectives and different hyperparameters within each objective. Fun fact, this distance matrix has about 2 million unique elements to it. And if you were to just do it with like a single for loop in serial, this would have taken about 10 years of compute time. But with some crafty parallelization, we were able to drop it down to a couple hours. So this actually shows how efficient our method is and how well it can scale. So with this distance matrix, because our method is a bona fide metric, we can, as before, visualize and embed each network into what we call shape space and color by objective. We can see that each color clusters together as we might hope, and that our method reveals representational differences between networks trained with different objectives. Alternatively, we can use non-parametric clustering analyses like K nearest neighbors directly on the distance matrix to reveal how network similarity relates to other variables of interest. But I'll go through that on the next slide. But before I go to that, there's only like 2 slides left, buddy. Does anyone have any questions? So as before, we can sweep the spectrum between covariance only and means only metrics to compute distance matrices at different values of alpha. Each of these distance matrices produces a different shape space embedding, as shown at the top. And if we were to do clustering, regression, or classification analyses directly on these distance matrices, I was able to find that we could networks position in shape space was very indicative of their reconstruction error on the reconstructed image for different values of alpha. Funnily enough, I was able to even decode with location in shape space what the random initial seed was. So like that integer where you set like torch.seed, whatever. And we were able to, if you're familiar with VAE literature, you'll know, maybe you've heard of disentanglement and how big of a mess it is. And basically there are like 30 different metrics or indices that people have proposed to measure what's known as disentanglement of the latent space of a VAE. And I won't go through all of them here. I don't expect you to take too much away from this plot, except that we can regress with using location in shape space disentanglement scores for basically every score. And one thing that I would like for you to take away is that by considering the noise, so like all the points that are not yellow, many times we do better than discarded in noise, which are the yellow points. So to summarize, quantifying representational similarity is a fundamental task in neuroscience and machine learning. Shape metrics offer a principled way to compare responses between many networks simultaneously. We develop here shape metrics for deterministic and stochastic neural responses. I've shown you that this can scale to millions of comparisons. And this allows us to directly relate differences in representational geometry to variables of interest. So with that, I'd like to thank you and my project collaborators. You can find our paper on the archive and the project code at this GitHub repo here. Thank you. And thanks especially to Nanda for inviting me.
So let's thank Lyndon for giving us a great talk today. So if there are any questions for Lyndon in person or online, I actually have a question myself. So Lyndon, if you could just talk a little bit more about the optimization with respect to T and how you ensure that it remains a rotation, because I don't recall there being any particular constraint in the objective that enforces this.
Right. Yeah. Sorry, did someone else say something? Right now. I think I'm hearing my echo, so I thought someone else had something. Let's go back to, let's see. Oh no. There we go. Okay. So in the deterministic case, if anyone is familiar with canonical correlations analysis, this is very familiar or very similar to that, in that we don't need to take like gradient steps or anything like that, or iteratively find T. We can just form the cross covariance matrix of the left guy and the right guy and take their SVD, discard the diagonal component, and like what is left, like the left singular vectors times the right singular vectors of that cross covariance matrix, is the optimal alignment immediately. So like that is solvable in closed analytic form. When it comes to the stochastic methods, it is a little bit more tricky because now we also have to consider this, the covariance as well. So if it were as before, like without any noise, then this would just simplify, as I've mentioned before. But since we have to take into account this covariance, there are a few options that we can take. One, and perhaps the most accessible, like using off-the-shelf methods, I should say, is if you're familiar with Riemannian manifold optimization. That's basically saying like, I want an object of optimization variable T. I want to do like gradient steps, but remain on, say, like the space of SO, or like the space of orthogonal matrices. So that is one option, doing literally like torch gradient descent and trying to remain on that manifold using Riemannian methods. We found that that was a little bit too slow. So instead of that, we reparameterized this Burus metric, and we developed like an alternating least squares procedure. And because this alternating least squares procedure is not convex, we just blasted like a bunch of random seeds, a bunch of random initializations, and just took the best one. Yeah, I think that's.
Yep, thanks. I think there's a question in the chat.
Is this method better able to cluster networks by their classification accuracy because discriminability is taken more into account relative to previous RSA methods? Yeah, that's an interesting question. I would say actually that RSA methods take into account discriminability more than this method, in that this method doesn't care how many stimuli there are, and doesn't care how discriminable each one is necessarily. It just really cares about like their overall relative geometry to each other. So I guess indirectly in there is discriminability, but I don't think there is an explicit accounting for discriminability here.
So we have a question in person.
Sure.
Thanks for the really interesting talk. I quite enjoyed that. As you're probably aware, there's also a lot of longitudinal recordings that experimentalists are doing now, where changes in the distance between representations or of the same stimuli are happening across time, and also changes in covariance of the representations that we're measuring or we think we're measuring. I was curious, in your code base and in this method that you're developing, do you actually return sort of measures of the distances and covariance-based like transforms that you're doing? So if neuroscientists want to quantify those things, they actually could? Or would that even be a meaningful measure in your opinion? For example, with representational drift and this phenomenon a lot of people are detecting across areas of the neocortex and hippocampus and these sorts of things.
Yeah, that's a good point. I would say that all the math presented here today is sort of agnostic to what you call network 1 and network 2. So network 1 can be time point 1, network 2 can be time point 2, for instance. You can measure the distance in their covariances. One thing that I did not show here today is this sort of difference in, let me see if I can. We have this interpolated distance of Wasserstein, and this basically decouples this contribution of means in red and just the covariance. So in the paper, we have like some experiments that look at, like, I have a whole bunch of untrained networks. You can think of this as like very early development or something. How related are their covariances now versus how related are their means right now? Then I have a whole bunch of trained networks. Let's look at the same thing. And we sort of propose this as a nice sort of diagnostic for like how well trained a VAE is in our example.
Right. Very cool. Thank you.
Thanks for that nice question. Thank you. Yes. So Alexander says, I'm still stuck on phi. That is very fair. Many people feel the same as you. The hypothesis space is quite large here, and there is no guarantee that a T will exist to relate the representations meaningfully. Can you validate the chosen phi in a post hoc fashion? Right. So I think the main concern with phi, let me go back to it so people can latch on to what I'm talking about here. The main concern with phi is that it is arbitrarily powerful. It can even, as I said before, be nonlinear. For the most part, we have just remarked that it can be nonlinear, and I have not done anything with nonlinear transforms, and I've tried to keep things as simple as possible. I think you're asking a more rigorous question of like, can I, in a post hoc fashion, sort of determine whether this phi was a good hypothesis or not? And I don't think I have a good answer for you for that, or I don't have a satisfying answer for you for that, unfortunately. So I think that would really depend on the questions that you're asking. And I would say that I have strived to just keep phi as simple as possible, like appending zeros or a simple PCA to match the dimensionality. Sure, thanks.
Do we have any other questions? Do you want to come here and maybe ask, I guess?
So this is with respect to the existence of transformation T. At the start of the presentation, you mentioned that they, the neural representations are in the same group. Is this a guaranteed condition every time, or like is this an assumption which you are taking while trying to get the metric function or the shape metric here?
Sure. So I think you cut out for a bit for me there. Did you say the neural representations must be, and then you cut out?
Come again, I didn't get you.
I guess, can you rephrase your question, please?
Oh, okay. It's like for the transformation to exist, they need to belong to the same group. How will is this a guaranteed fact that they belong to the same group? I didn't understand that. It was mentioned at the start of the slide, so.
Right. Sure. No, that's a good question. So this basically has to is related to, where is it? Is related to what Nanda was asking, in that, like, how do I ensure T always stays in the same group? And I think this is an optimization detail, and there are many options and different paths that you can take. One that we took was a very fast sort of SVD approach, where we guarantee that T will always be an orthogonal matrix, and that helps. If you say want it to be a permutation, then with each to align them, you can solve like a simple, like off-the-shelf linear systems, sort of like a Hungarian algorithm sort of approach, and that will also guarantee that you get a permutation. There's some caveats to that, but for the most part, that's true. Thank you.
So if there are no more questions, I'd just like to thank Lyndon again for the great talk.